So, good afternoon to everyone, and welcome to this first workshop on spatial transcriptomics and cell-cell communication modeling, new opportunities to study the cellular dynamics of biological systems. So, my name is Giacomo Baruzzo, and today with Enrica Calura from the University of Padova, Luz Garcia Alonso from the Wellcome Sanger Institute, and Gabriele Sales from the University of Padova, we are going to chair this event. So, first of all, how you can interact with us? There are two main ways. There is a live Q&A chat that you can find on the bottom of your screen, where you can type any question that you want to the speaker. During the speaker presentation and at the end of speaker presentation, we will try to read your question to the speaker such that they can answer immediately at the end of the presentation, during the entire event, or in the final Q&A session. You also have the opportunity to discuss with other people, all the other attendees of this workshop, using the discussion forum that still you can find on the bottom of your screen. As you saw from the website, we have a schedule full of talk. We have 7 invited talk and 4 contributed talk, all on the topic of spatial transcriptomics and cell-cell communication modeling. So I will not stall any other time, since the schedule is quite full of talk, and I will leave the stage to Luz, that is going to introduce the first speaker, Elisabetta. So please, Luz, the stage is yours.
Thank you, Giacomo, for leading the organization of the first workshop. And thank you, everyone that joined today. So our first speaker is Elisabetta, and she's a group leader at the Cellular Systems Genomic Group in the Josep Carreras Research Center in Barcelona, where she uses single-cell and spatial technologies to study inflammation. So, Elisabetta, please, whenever you want.
Hi, everyone. Just 1 second that I'm sharing the screen. Okay. So, can you see it?
No.
No? Yes.
Okay.
Yes, sure. Okay. Yeah, 1 second. Now? Okay. Okay. So, hi everyone. I hope that you can hear me properly. Can you confirm, please? Oh, okay, perfect. So, thanks for the kind introduction and for the invitation to this nice event. Today I'm gonna talk about the work that I'm gonna introduce the work that we are doing about benchmarking of spatial transcriptomics tools with the aim to accelerate precision medicine. So first of all, I will explain briefly what we do in the lab, so to put you in the context. So basically, in my lab, we are interested in the characterization of complex tissues at molecular and cellular level. In line with the Human Cell Atlas project, we combine single-cell technologies and spatial transcriptomics in order to obtain molecular and spatial reference maps of the normal tissues. Given that these technologies are still imperfect and the data that are generated as well, also the technologies is in continuous evolution, technology benchmarking is fundamental in order to make sure that what we measure is biological and not a technical artifact. So this type of characterization is particularly interesting when cells are affected by natural perturbations, for example, such as aging or human diseases. Therefore, in this context, we are interested in the characterization of inflammatory process that recur in this perturbation. So in this context, Spatial transcriptomics represents a powerful tool to explore tissue structures. And recently, these technologies have exploited several types of tissues, and here I'm just putting some of these examples. For example, recently I've been published this paper in which with merfish, the mouse primary motor cortex have been characterized in order to profile spatial cell type profiling. Also with the StereoSeq, we obtained a spatial temporal characterization of a salamander brain, and particularly authors focused on the development of the brain and regeneration after injuries. Also, this with Visium and its integration with in situ sequencing, we could obtain a spatial temporal characterization of the developing human heart. And more recently, spatial transcriptomics have been used to profile myocardial infarction. So it's becoming always more clear that this type of technologies can be very useful, not only to answer questions in fundamental biology, but also can have an impact in clinical applications. Mainly, we can recognize two broad classes of technologies. One of them is the imaging-based spatial transcriptomics, are the imaging-based spatial transcriptomics methods, which are targeted and enable the in situ transcriptomic profiling at molecular and single-cell resolution, and then the array-based spatial transcriptomics methods, which are untargeted and are typically are obtained by using an RNA capture method, followed by sequencing at spot resolution. So in this case, we lose the single cell resolution, and because the spots are basically barcoded, spatially barcoded microarrays that might contain up to 10 to 15 cells. Therefore, this of course shapes also the types of projects that researchers are applying recently. Research is in fact often based on a two-steps approach, where first we formulate an hypothesis using the untargeted high-throughput technology, and then we test and validate our data-driven hypothesis with the imaging-based technologies. However, also with the implementation of new tools, analytical tools that have been recently implemented, we can also try to improve the resolution of this data, or improve the RNA transcript detection. Therefore, a number of tools have been recently developed in the last 2 years, and here in this plot, you can see how the cumulative number of published methods according to the category of analysis that they were developed for. So from here, in my lab, we were interested in make a more clear identification of this analytical task for spatial transcriptomics analysis. And we have therefore identified more or less 5 classes, which correspond to each analytical tools. With each analytical task. So first of all, the sensitive cell type mapping, which is the ability to define the exact location of the cell population, including rare subtypes. Second, the accurate tissue patterning, that is the ability to detect fine tissue microstructures, enabling the interrogation of distinct gene patterns. Transcript imputation and the integration of spatial transcriptomics with images, as well as harmonization of integrability with the single-cell RNA-seq and co-registered imaging data, which enable the high-resolution molecular and histological mapping of tissue sections, and then a cell-cell interaction that is important to characterize to understand the organization of the tissue and therefore is functioning. So if we if we group these tools according to their analytical task, we found these different categories. And in our lab, we attempt to have a first qualitative comparison. Our analysis bring us to these results. So we found out that computational spatial transcriptomics tools differ substantially in their performance, and main differences are in their ability to identify distinct spatial gene expression patterns, link cellular and functional phenotypes to structural architecture, characterize morphological variability in histological images co-registered with spatial transcriptomics data, and also infer intercellular crosstalks. These results were also partially observed in a recent benchmarking of spatial methods, in which the authors focused more on methods for integration of spatial transcriptomics and single-cell RNA sequencing data, particularly for the task of the prediction of the spatial distribution of transcripts, and then a cell type deconvolution. In this paper, the authors used several published methods and dataset, spatial transcriptomics dataset, together with a single-cell RNA-seq data, to compare the different approaches. And also they simulated the dataset in order to have a ground truth that was also obtained by downsampling the original data. The downsampling was done randomly, just removing reads from the original data. And then in order to compare the expression of the detected transcripts from each tool. In second instance, they were interested in measuring the accuracy of predicting cell type deconvolution. And to do so, they generated these synthetic spots by combining several cells and using these spatial grids on top of spatial transcriptomics map. And in our case, we want in somehow extend this type of benchmarking by generating a new flexible framework to generate a spatial transcriptomics ground truth dataset. Our framework will consist of two main steps. One step is we'll start with the use of a high-resolution spatial transcriptomics image that is used as the basis to simulate a spatial grid of spots that will represent an ideal pseudovisium dataset, where gene expression patterns are enhanced. This dataset will be called iVisium. And then a noise model is learned and applied to this iVisium data in order to downsample the data strategically and obtain a realistic pseudovisium dataset. So the idea behind the noise model would be, so we will use several strategies, and still we are working on that. But the idea behind that would be to use a model that will allow us to obtain a discriminator that will be able to provide a feedback to the generator in order to simulate as much as possible and with a high accuracy an original Visium. This type of approach will allow us to employ our framework in distinct analytical tasks and also under distinct biological settings. For example, here I'm showing just a few examples: spatial gene expression patterns, mapping cell types to spatial tissue structures, and inferring cell-cell communication, and also the other tasks that we have discussed previously. As a first instance, we tried with the parametric downsampling strategy, and also we are testing the model as a proof of concept with brain dataset, because these are well characterized tissues, and also a lot of datasets are already available in both high resolution spatial transcriptomics data, but also in a Visium dataset. So our downsampling strategy at the beginning was meant to mimic the experimental steps that and the main characteristics of high throughput spatial transcriptomics data. Therefore, we have considered parameters that allowed us to include dropout that follow the typical distribution in both spatial transcriptomics but also single cell data. However, when we measure when we compared our synthetic data Visium with the original Visium, we found out that while the mean and the coefficient of variation was quite consistent, in the case of the variance, we were observing much more variation in our Visium than in original. So that led us to understand that probably a deep learning approach would be, such as the generative adversarial network, can be more suitable to better simulate a Visium dataset, the Visium distribution. Also, we evaluated gene expression patterns, distinct gene expression patterns in our R-Visium compared with the I-Visium, which would represent the ground truth data, and also the MERFISH. And we observed that, for example, in the case we have here, for example, okay, thanks. Here we have, for example, a pattern in which genes, the distribution of the genes is localized in this area or around this morphological structure, while here we have a more sparse distribution. By doing a fast correlation analysis, we found out that these relationships are conserved in our R-Visium with the original I-Visium, while the linear correlation is a bit gets a bit lost, except that is a bit is more maintained in the context of genes that are localized in a specific area. So now with our spatial transcriptomics framework, benchmarking framework, we can compare, for example, different tools, for example, denoising, cell type deconvolution, or cell type co-localization. Of course, this is just the first attempt, and also we have to work in our deep learning downsampling approach to better improve our approximation. But now the work is still ongoing. So just to sum up the work and the information I gave in this talk, I want just to say that high-throughput spatial transcriptomics technologies are limited by low spatial resolution and capture efficiencies. Therefore, it's important the application of powerful computational methods to denoise the data and obtain optimal tissue maps. And also, spatial transcriptomics tools differ substantially in their performance, and this is clear in distinct tasks and biological scenarios. Therefore, benchmarking spatial transcriptomics tools is fundamental and will define a set of guidelines and quality metrics. And this would help researchers to select the best methods according to their biological needs. Also, this requires the generation of ground truth dataset with distinct biological properties and in order to make a comprehensive evaluation of the tools. And now I just introduced our flexible, our idea about a flexible framework that will result in the generation of realistic ground truth dataset that can be used to perform large-scale comparative analysis of the most common spatial transcriptomics methods in different tasks and under distinct biological settings. Thanks a lot for the attention, and I will be happy to take any question.
Hello, everyone. Any question for Elisabetta? I'm not sure whether I understand how this works. So questions should be in the live Q&C or the discussion forum. Am I right? Okay. Well, I have a question for you. And yeah, so I've never tried to do this before, but I'm always wondering, what are your thoughts about mapping immune cells in VSU? And I'm saying this because immune cells are quite rare in the spots, so it will apply to any rare cell type, actually.
Yeah,
Okay,
that's...
Do you have any advice to anyone trying to do this?
Yeah, that's very challenging, and I have to say that now there are always more tools every day, every week, so we are testing them, and let's say that immunology is not is not super easy. It's much more challenging than the identification of immune cells in the histology. It can be really hard, and so it's really something that we want to test in our benchmarking, because still I think that we don't have a good ground truth data set to make this test. But yes, we have tried some tools, and there are some that are very good. Particularly, for example, I like the XFuse, for example, or and also base space, that seems to be interesting. And but this really depends on the pattern of the immune cell types or the gene expression that they express, if they are more sparse or if they are colocalizing in a specific area of the tissue. So it really depends on the situation of the context. That's why we really, really want with this benchmarking to provide a ground truth data set that will allow data to test these different situations.
Okay, sorry, I don't know who can hear you. So yeah, let's move to the next speaker, and I think now it will be Giacomo introducing it.
Yeah. Thank you, Luz. So our next speaker is Lucas Weber from the John Hopkins Bloomberg School of Public Health, that is going to have a talk about a method to have a scalable identification of spatially variable genes in the framework of Bioconductor. So please, Lucas, the stage is yours.
screen. If that's not sharing correctly, could you please let me know? Everything's fine? Okay, that's great. Okay, so I'm going to present work that we've been doing on a new method to identify spatially variable genes in a scalable manner in spatially resolved transcriptomics data called NNSVG, and we've implemented this within the Bioconductor framework. So first, some quick background. So we've been working mainly with the 10x Genomics Visium platform, which measures transcriptome-wide gene expression at spatial resolution by having a grid of spatial locations on a tissue slide, and then the mRNAs are captured and barcoded and sequenced from each of those spatial locations. So the dimensions here are small tissue slides of about 6.5 by 6.5 millimeters with 5,000 spatial locations or spots. And then, yeah, we measure transcriptome-wide expression at each of those spots and do analyses such as identifying spatial domains, spatially distributed cell populations, and spatially variable genes, which is what we've been working on. And also showing an overview of what the data looks like. So we have gene by cell expression count matrices similar to single cell. Now in the spatial context, it's genes by spatial locations. Then in addition to that, we have the spatial coordinates of where the measurements come from, which I've illustrated there. And we can also get image features, like morphological features, cell features. In this work here, we're using the expression counts and the spatial coordinates, which is what I've circled there in red. Now, spatially variable genes. So here we are interested in identifying genes that are associated with spatially resolved expression patterns on the tissue slide. So in this example here, this is from the dorsolateral prefrontal cortex brain region in the human brain. In this study, I'm showing one Visium sample there 6 times, so showing expression of 6 different genes. And in this brain region, there's this layer structure. So we're interested in identifying genes that are associated with spatial patterns, such as those layers, which is the top 3 example genes there, but also any genes associated with other structures, other spatially resolved structures within the same tissue slide as well. And the 3 at the bottom there are examples of those. They're associated with blood and immune processes. And what's really important here for a method to identify these genes is that these different types of genes can have quite different spatial ranges in their expression patterns, which is what I've annotated there with the red arrows. So some of them have quite large patterns and others small patterns, and we want to identify all of these. And why are we interested in spatially variable genes? So for a few tasks, so one is data pre-processing, so we can reduce the number of genes to a smaller set of biologically informative genes and use that for further downstream analyses. Secondly, also simply to get a top list of top informative genes to investigate individually. And then the question becomes, how do we define biologically informative? And in the non-spatial world, we often refer to highly variable genes. Now in the spatial context, that becomes spatially variable genes. That refers to different methods. And also showing here how this fits into analysis workflows. So this spatially variable genes fits into the feature selection step, either for reducing dimensionality, as I just mentioned, or for identifying top-ranked spatially variable genes. And yeah, so I mentioned there are different methods for identifying these types of genes. So in non-spatial data sets and single cell data sets, common methodology is called highly variable genes. This ranks genes by what's referred to as excess biological variation above an assumed technical trend, which accounts for a mean variance relationship. Then you can take, say, the top 10% for further downstream analyses. Now in spatial data, we also want to take the spatial information into account. The simplest method there is to use what's called Moran's I statistic that can be used to rank genes by their spatial autocorrelation. But now more recently, new methods have been developed that are specifically adapted to spatial transcriptomics or spatially resolved transcriptomics data. And that's where we refer to spatially variable genes and methods to identify them. And these are some examples of methods that have been published recently within the last 5 years or so to identify spatially variable genes. 3 here, so Spatial D, Spark, and Spark X. One of the first ones was Spatial D by Svensson et al. 2018. This uses Gaussian process regression using a spatial covariance function and kernel on distances, and then a likelihood ratio test. This was a very nice method, but scales cubically computationally in terms of the number of spatial locations, which was fine at the time with a few hundred spatial locations, but in the newer Visium platform and even newer ones coming along in the future, we have a lot more spatial locations per slide. So in Visium, it's sort of 3,000 to 5,000 spatial locations per slide, and in the future with other platforms, it'll be even more. Also, SlideSeek has a lot more. So scaling cubically is just too slow then in the end. So we were inspired by that and interested in developing a new method to identify spatially variable genes in a computationally scalable manner. So in the statistics literature, there are a few ways to improve computational scalability in these types of models. One approach is called nearest neighbor Gaussian processes. This is the one that we ended up using. This was introduced by Data et al. In 2016, one of our colleagues at Johns Hopkins. This uses an approximate likelihood from Vecchia, 1988, with a small set of nearest neighbors, like 10 to 15 nearest neighbors out of the thousands of spots that you've got. And that approximates the data very well and gives you a sparse precision matrix, the inverse of the covariance matrix. And that ends up letting you get down to linear scalability in the number of spatial locations, because it's due to the inversion of that full covariance matrix previously, that's cubic in the number of spatial locations. But with this sparse precision matrix, that becomes linear, which is a huge improvement in terms of runtime. And they have two very nice R packages there, SPNNGP and BRISC. We've built on BRISC in the NNSVG framework. This fits one model per gene using BRISC, which, so I've written the model there. We assume an exponential covariance function, not a squared exponential, and that has a gene-specific length scale parameter, which that links back to those red arrows that I had on the initial example slide. And then we also calculate a likelihood ratio statistic, which we can use to rank genes, and likelihood ratio test to determine significant genes. So yeah, the key points there are that this becomes linear in the number of spatial locations due to the use of the NNGP. And at the same time, we still have a gene-specific length scale parameter fitted independently per gene. We can also include covariates for spatial domains. And we've implemented this. It takes about 45 minutes to run on a laptop or faster on a high-performance cluster with more cores. Now I'll show quickly some evaluations. So we've evaluated this on several data sets from the Visium and other platforms. In this example here, this is the same data set from our previous work with our collaborators from the human dorsolateral prefrontal cortex. On the bottom left there, I'm showing ground truth annotation for the layers, which is the structures that we're interested in. And on the right, I'm showing again those same 6 genes that I had at the start. The top 3 are associated with the layers, and the bottom 3 are associated with blood and immune processes that have different ranges in their expression. And here I'm showing, so we run an NSVG on this data set, and here I'm extracting the fitted length scale parameters. And this is as a check to see that if those length scale parameters are sensible for these 6 specific genes that we're highly interested in. And yes, so we get very small estimated length scale parameters for the 3 at the bottom, HBB, IGKC, and PY, which is as expected, and larger ones for the ones at the top, which have the larger ranges in their expression. So that's good. And then for one of our evaluations here, we're still focusing on these 6 genes. I have more genes coming up later as well. But here, for these 6, which we know are very biologically informative spatially variable genes in this data set, we evaluate this by calculating the ranks that we get in the list of all spatially variable genes from this data set. Where do these 6 turn out in the rankings? And we do this so we want them to be highly ranked from our method. We do this for NNSVG, which is our method, and also for SparkX, which is an alternative method using a different mathematical framework that's also linear in the number of spatial locations. Then we also compare against HVGs as a non-spatial baseline method and Moran's I as a spatial baseline method. And we see that all 6 of these genes come out as quite highly ranked with NNSVG and also with HVGs in this data set, and not with the other competing methods. So that's good. And we also we have several other data sets where we do similar evaluations using other genes that are known to be informative in each data set. Here, this is an additional extended set of biologically informative spatially variable genes in this data set. This is 137 genes associated with the layers. And we show here, I'm comparing the likelihood ratio statistic against the rank, and 136 out of those 137 come out as significant, which is also good. And I'm highlighting those 6 on there as well. Then we also simply plot the top genes that come out from our method and the competing methods. Here on the left, it's the top 20 genes from NNSVG, and on the right, the top 20 genes from SparkX. And we see in the ones from NNSVG on the left that most of those top 20 genes, top 20 SVGs, are associated with the distinction between white matter and gray matter, which is the red and black in the ground truth annotation in the top right panel. And that's good because we know that in this data set, the strongest biological signal there is white matter versus gray matter. So we expect that most of the top genes should be associated with white matter versus gray matter, and they are in these plots from NNSVG. And then we also do some simulations to confirm that it really is linear in the number of spatial locations. So here I'm downsampling 2 data sets, the Visium one on the left, a different one from SlideSeq on the right with more spatial locations. So I'm just downsampling the number of spots and then running it 10 times. So I'm showing that it does scale linearly with the number of spots. Okay, so a couple minutes left. I'll mention quickly on the implementation. So we've implemented this as an R package within the Bioconductor framework. Inputs and outputs can either be spatial experiment, which is a structure we've developed in the Bioconductor framework, or they can also be simple numeric matrices. We've done this all within Bioconductor to facilitate building it into analysis workflows, into modular analysis workflows. And the package can be installed from Bioconductor or from GitHub. There's a vignette and tutorial there as well. And we have a preprint up. It's currently going through revisions. And then I wanted to just mention a bit more about Bioconductor. So Bioconductor is a large open source community-based software project for high throughput genomic analysis using R. There's more than 2,000 contributed software packages there, and a really nice, well-maintained core infrastructure such as data structures and documentation and testing infrastructure. So we've developed within that framework, and we use the spatial experiment data structure. This is work that we've done together with also Helena Kroll, who's speaking later today as well, and Dario Righelli. This is a data structure we've developed to store spatially resolved transcriptomics data within Bioconductor. It extends single cell experiment, which was previously developed for single cell data, and includes additional slots such as spatial coordinates and image data. That's also described in this paper that we've published and can be installed from Bioconductor. And in ongoing work, we're now also developing some example analysis workflows in an online book called OSTA, Orchestrating Spatially Resolved Transcriptomics Analysis with Bioconductor. This is a work in progress, but the draft version currently is online. So we use example data sets, and then there's sort of interactive R code and examples to show how to do analysis workflows within Bioconductor for these data sets. So in summary, I've presented our new method called NNSVG for scalable identification of spatially variable genes in spatially resolved transcriptomics data, which builds on the nearest neighbor Gaussian process framework and the BRISC R package to apply this. It's linearly scalable in the number of spots and fits a flexible length scale parameter, unlike other existing methods. Optionally also covariates for spatial domains. we evaluated on several different data sets in our paper. In our preprint there, I showed one of those data sets just now, the Visium one. And yes, there's a preprint online. And then in addition to that, I showed the spatial experiment structure within Bioconductor and our ongoing work on Oster analysis workflows. And with that, that's my acknowledgements. So thanks to my advisors and colleagues and collaborators and our funding there as well. Thank you. So I'll unshare.
So thanks, Lucas, for the very nice talk. So there are questions from the audience. Otherwise, I have a few of them. So let me check. So there is a question that comes from Ruth, if I got it right, the nickname in the chat. So it say, you have nicely show how different methods and your NNSBG method to select spatial informative genes do for marker of major lineage in the brain. Going back to my observation on rare cell type, how you evaluate how methods do with marker or rare cell types on the brain?
So rare cell types. Yeah, so I guess, so yeah, we could have spatially variable genes that are associated with just a very small number of spots. And we do have that actually in the data set that I showed there, those where I kept showing that panel of 6 genes, the 3 at the bottom had the quite small patterns. They were associated with just a tiny region of blood and immune cells. So yes, so I think our method will work because we have that flexible length scale parameter that can even have very small ranges in expression. I guess, yeah, in terms of quantifying rare, I'm just thinking in terms of the number of spots. I don't necessarily know if that's going to be a cell type because we're just looking at spot level. So that kind of partially answers the question, I guess. If it's a rare number of spots, it should work, and our evaluations show that it works for those specific genes there.
Okay, so I see that there are many other questions in the chat. Probably we have time for another one, then as usual for you and for the other presenter, you have a chance to answer directly in the chat and in the last 15 minutes, but I think that we have time for another question. And one is, say, thank you for the great and comprehensive talk. Do you think it would be feasible to run NNSBG computation on GPU computing to have a even further speed up?
Yeah, so GPU. So I've implemented it to be parallelized on CPUs. So yeah, if you have lots of cores, so it's literally just fitting one model per gene. So if you have thousands of models, more cores is faster. I haven't implemented it to take advantage of GPUs. Yeah, and off the top of my head, I wouldn't know how to extend that in that direction either. But it's parallelized for CPUs. So yeah, that partly answers.
Great. So I would say thanks again to Lucas and even to all the people in the chat that do the questions. So again, Lucas will try to answer in the rest of the event. And again, some question will be even selected for the final 15 minutes discussion panel. So thanks again, Lucas. And now I leave the stage to Gabriele that will introduce the next speaker.
So thank you, Giacomo. Next up is Giovanni Pala from the Helmholtz Center in Munich, who will be talking about how to use Python actually to handle spatial and multimodal data. So the stage is yours.
Thank you very much. You can hear me and see the screen, correct? Okay, thank you very much. Thank you to the organizers for the invitation. Indeed, I'm going to present works on spatial data, a new data format, and Python object that we've been working on lately. I should mention this is a collaboration with several people, in particular Luca Marconato from EMBL, Kevin Yamuchi from ETH, Isaac Bierschup and Fabian Theis from Helmholtz, Josh Moore from University of Dundee, and Oliver Segre from EMBL. So I'll briefly go through the table of contents. So I'm going to introduce and motivate spatial omics analysis with previous work we've been doing on Squidbey. and then list some of the limitations that we had with the Squidbey framework for handling spatial omics data, both in terms of storage and manipulation. And then I'll talk about the project's spatial data and end up with conclusions and perspectives. So first off, so how does the single-cell analysis ecosystem in Python look like? It started, let's say, with ScanPy, probably the most popular tool for single-cell analysis in Python. And many other tools have followed that deal with the different aspects of analysis of single-cell multimodal data. Examples of these are Muon for multimodal omics analysis, SCVI for probabilistic models, in particular variation autoencoders, cell ranks for probabilistic fate mapping using RNA velocity, and CIRPy for immune cell receptor signaling tools. These are some of the examples that are part of the single-cell ecosystem in Python, and again, deal with different aspects of the data analysis. And what they all share is the common data representations, namely UNData for single modalities and MU-data for multimodal data. So there has clearly been an increasing need of spatial transcriptomics, so computational tools for spatial transcriptomics data analysis. Recent review named from Nature Methods named Spatial Result Transcriptomics Technology of the Year. And there is a lot of infrastructure in Python for dealing with some of the modalities that spatial data sets are made of, essentially. So this was really the motivation for Squidbey. For Squidbey, we wanted to sort of make a bridge between what we think is available in Python for dealing with such data and the single-cell analysis ecosystem. So meaning specifically using UNData and other type of data representations for the analysis of such data. I also want to mention briefly that this gray box here that entails these different packages and many others actually now has a name. We launched an organization called SCVerse. With SCVerse, we're essentially trying to put together foundational data structures and tools for analysis of single-cell data mostly, but maybe in the future for more generally life sciences. Again, leveraging state-of-the-art frameworks for scientific computing in Python and allowing both users to use these tools and method developers to build on top of these tools to deal with different problems and challenges in data analysis. So Squidbey, in a nutshell, the motivation was, again, that when we talk about spatial omics, spatial technologies, and this has been very nicely shown also by previous speakers, we actually mean a bunch of different measurements. Some of them are sequencing-based, some others are based on imaging, some others are based on mass spectrometry. So there is a wide variety, and there is a lot of tooling that goes through the processing and QC of such data. With Squidbey, we kind of wanted to go more towards downstream. So after this data has been processed and QC'd, what's the type of data structures analysis the user might want to be able to have and do for the analysis of such data. And we abstract, let's say, two main new objects, the spatial graphs, which is just a nearest neighbor graph, the same that we're used to use as a backend in many single-cell analysis. But now it's a nearest neighbor graph based on the spatial coordinates, and then an image container, which is essentially aims to store the microscopy images, which are often acquired. And then also in Squidbey, we provide a bunch of downstream analysis tools in terms of exploratory statistics, like exploratory data analysis and summary statistics for analyzing, for instance, spatial communities, ligand-receptor interactions, types of extracting various types of spatial patterns, as well as more towards the image analysis side and interactive visualizations. And we really strive to make all of these ready to be interfaced with the rest of the ecosystem in Python, in particular around machine learning and deep learning. So on the spatial graph side and spatial statistics, what we have essentially is we provide different ways to build the spatial graph, because again, according to the technology, there are different ways that the user might want, the analyst might want to encode spatial similarities in a graph representation. And then again, analysis that leverages these representations, such as the neighborhood enrichment. In this case, we're looking at a neighborhood enrichment score across different clusters for a section of the mouse gastrulation. And we show that we recapitulate some of the clusters that have been found to be spatially enriched by the original authors. And then on the image analysis side, again, as I mentioned, we sort of introduce this image container, which is a thin wrapper of X-array, so for working with large arrays, which is sort of implements some methods for pre-processing, segmentations, and also feature extraction. We really believe that the image that is acquired, let's focus now maybe on spatial transcriptomics, namely Visiums and others, maybe nanostring, should be analyzed as a modality, because it can be a lot of information can be extracted from, for instance, the morphology and so on. And then again, we provide downstream interactive visualizations via an Apari plugin. One thing that we really try to do was to be able to also relate it to the undata. So be able to essentially, to a very sort of coarse level, subset the image or the undata and propagate this subsetting and cropping between the two representations. So between the pixel level representations and the undata, the table representations. Now, there were, since really from the beginning, we realized that there were many limitations on this approach. And here I'm listing some of the maybe most obvious ones. So in particular, let's say starting with fish-based data, we do not really have a way to store, analyze, and visualize the molecular level representation. What do I mean? In fish-based data, we often, the data types often a very long list of spatial coordinates where each of the transcripts has been measured, so each of the gene. And the user might want to be able to aggregate these transcripts in different ways, going back and forth again on the analysis pipeline, and none of this is possible really from this type of approach. The microscopy images were mostly dealt by the image container, but this is a suboptimal solution because first off, it's a separate objects, and it's a bit unclear how the user would essentially relate these two data sets, the molecular level, let's say the undata level, where gene expressions or protein expressions is stored, and the original pixel image. There is no support for coordinate transformations. This is, again, very important feature because oftentimes images are acquired across maybe different stacks, different samples, maybe the same samples, but on different regions of the samples. So it's important to have ways to relate these different coordinate spaces. Also, sometimes the images are processed, such as registration, and there are transformations that are learned from such process. And again, as I mentioned before, we did try to be able to relate the pixel level representation of the image with the undata level representation, but we really didn't go very far. And many of the operations that are actually useful, such as, again, the aggregation I mentioned, the cropping and subsetting and rasterizations are not available. And, oops, sorry, I went back. So this is why we started to work with spatial data with a core team that I mentioned before. And so the idea of spatial data is to store, first off, to have a way to store the multimodal, different multimodal views of the spatialomics data set in a very efficient way. And also, most importantly, using a community-driven specification, which is the NGFF specifications developed by the OME organizations that's been for many years involved in developing standards for image data formats. And then on top of such storage representation, we wanted to build a Python representation in this case, which could essentially allow the user to access such data in an efficient way, to stream such data for the machine learning and sequence analysis tools that we are used to, and also for visualization. For visualization, both in Python for the interactive visualization of image data with an Apari plugin, which was kind of ported from Squidbey, but also to be able to share it, maybe via cloud-based access, such as with tooling such as Vitessic, which allows visualization of on the browser of very large images. So how spatial data looks like? So the core of spatial data is made of elements, what we call elements or building blocks. And these building blocks are essentially trying to encode the data types that the spatial data set can be made of. So there are the first two main layers are images and labels, and both of them are essentially raster images, so pixel images. One of them contains the intensity, the colors, let's say, and one is a segmentation mask representations. And this can be really, ND arrays, can be 3D, can be 4D, according to the type of technology and measurement that has been done. Then we have points, and point stores, for instance, the transcript location. So essentially like a point cloud of spatial coordinates that can also be spatial index for doing some type of operation such as the aggregation in a very fast and efficient way. Then we have polygons, which stores essentially more general type of shapes and regions. And this can be both spots, for instance, or spots in the case of like DeepSeek or nanostrings, where we have maybe regular regions, but they can also be like anatomical regions, for instance, in the case of the brain, which are usually annotated and can be easily defined. And then we have tables, and tables are really essentially the let's say the bridge or better, the elements that tries to bridge the imaging world with the sequencing world. Tables are essentially annotate these building blocks by storing the annotations such as cell type, clusters, metadata of various kinds, experimental covariates, and so on and so forth, and also gene expression, protein expression, etc. And as a logo, I put the undata because tables can be an undata. So the idea is that you can have an undata that annotates all of these building blocks. So in terms of API, this is how it looks like. So as I mentioned, we have a storage defined by the NGFF specifications and implemented in ZAR. So it's essentially a ZAR container, a ZAR store, and we would have a sort of very collections of these building blocks, which we call the spatial data container, which essentially just a listing of what is present in this data file. And then the user would decide which type of building blocks to combine in a spatial data object, which provides the type of analysis that I'm about to mention. And these spatial data are really the sort of the actionable, the object that the user uses for the analysis. So on the operations, as I mentioned, we want to be able to combine these building blocks in different ways. So some of the operations that we are prioritizing, these are the main ones, is the cropping and subsetting. So essentially cropping, let's say, a region of the image and then propagating such crop also to the table. So essentially say, I want the top left of the image and I automatically getting all the observations that are present in the top part of the image, but also the other way around. Let's say I want to subset all the cells by specific cell types and getting all the regions in the image or polygons and so on and so forth that are belong to that. Rasterization, so essentially going back, rasterizing, for instance, points or polygons is mostly useful for visualization. And then aggregation, such as I mentioned, counting the number of transcripts under each cell, for instance. This is useful for fish-based data. And then lastly, the coordinate spaces. So coordinate spaces can be seen in two ways. So one is essentially when we have a local and a global coordinate space. Maybe we have coordinate spaces that are the pixels of the image, and then a global that are the actual micrometers. So where this pixel image has been measured on the tissue. And we want to be able to have also transformations that are able to transform between these spaces. But also they can be results of maybe analysis, such as affine transformations. The way we implement them are essentially stored as dictionary essentially, where we have a name coordinate space and a name axis and the type of such axis. And then the name of the coordinate transformation between these spaces. So if we have pixel space and micrometers, we want to be able to go from pixels to micrometers and from micrometers to pixels. I should mention that this work has been done mostly by John Bogovic on the NGFF sides, again on the specifications, and we are relying on the type of specifications that the community agreed on. And then finally, with this I'm finishing my talk, the visualization and ecosystem. So as I mentioned, especially as part of the SCVerse ecosystem, so we really want to make it available and be able to be used by the SCVerse ecosystems and again by developers of method developers of the ecosystem. And on the visualization side, because we natively use ONGFF and ZAR, we will be able to allow to visualize such data also from other type of visualization tools. So not only Python for Knapari, but for instance also Vitesi for JavaScript-based tool on the web, which can already read ZAR, and then the Java equivalent for Mobi, called Mobi. So to summarize, spatial data is a fair framework for multimodal spatial omics data. It builds on community-driven specifications and implementations to enable cross-platform analysis. It leverages state-of-the-art frameworks in Python to operate on large arrays. It can store multiple modalities and multiple data types to accommodate the wide diversity of spatial omics data. It can perform operations between elements as well as transformations, and it can be plugged in a variety of visualization tools. So I should mention this is very much ongoing work. There is not a release candidate yet. We hope to have a release candidate by end of 2022. I just want to mention that this is the let's say the roadmap. So thanks to collaborators, again, mentioning the core team and many other contributors that have been working on this. We have all the conversations on the development of tools in the public. So SCVerse, Zulip Chat, ImageSC, Zulip Chat, and we have BWiki call. You can join Zulip to if you want to attend. I very briefly want to mention that this started as the very first post-pandemic hackathon in April 2022. And it went on with a series of other hackathons with people, so it's been very fun to collaborate again in person. And with this, I want to finish, and thank you very much for the attention, again, to the organizers and sponsors.
So thank you so much, Giovanni, for this nice walkthrough of all the features that this new framework offers. So let me check if we have in the Q&A some questions. I don't see any question for the moment, so let me just break the ice here with one of my own. I was wondering, I saw that you're using the ZAR framework as a starting point for storing your data. And since all of the things that you were talking about are likely to be quite large as a footprint, both in memory and possibly on disk, I was wondering whether the ZAR framework provides some on-the-fly compression, so to reduce the amount of space that is required.
Exactly. So the motivation for using ZAR and then as a storage framework, and then X-ray and DASKs to operate on these large arrays, which indeed can be quite large, right? Several gigabytes for each of the images, and most of the times you don't want to read the whole image in memory. Maybe you don't even want to read the whole image on your local, and you want to access it again via cloud-based access, maybe an S3 bucket and so on. And because of the size, we need to rely on what's the state-of-the-art in Python for accessing these large arrays and enabling lazy operations and delayed operations. So the motivation for that is exactly this, because it allows to do this type of, again, lazy operations and delayed operations on the array.
Okay, so thank you again for your talk, and let me leave the stage now to Giacomo, who will be introducing the next speaker.
So thank you, Gabriele. The next speaker will be Elena Crowell from the University of Zurich with a talk about how you can use a spatial single-cell imaging platform to profile, in this case, inflammatory bowel disease. So please, Elena, the stage is yours.
Hi, thanks for having me. Oh, I can hear myself. Any advice what I can do? Yes, can you still hear me? I cannot hear it, right, but yes, I assume. Okay, my apologies. Okay, thanks for having me. And just to start with a quick acknowledgement that I'm from the University of Zurich, but I was recently visiting in Barcelona. And this is joint work with the Holger Hayen Lab and Azucena Salas at the IBD Lab and two PhD students. And the data is kind of early access from nanostring COSMX data. And this is a targeted imaging-based spatial transcriptomics technology. Targeted meaning we're not measuring the full transcriptome, but in this case, 1,000 features approximately, which is still a lot more than early Murfish studies, etc. And because it's imaging based, we have molecule level resolution. So we have spatial coordinates for both molecules and cells, given we have segmented our cells. So the experimental design is the following. We have 9 column sections across 3 experimental conditions, 3 healthy controls, 3 Crohn's disease patients, and 3 ulcerative colitis. And these 2 disease kind of groups are different levels of inflammation. So even within each of these, there's differences in how the disease manifests. Per section, we have about 20 FOVs. These are field of views. I'll show you what that looks like. Per field of view, we have about 50,000 cells, amounting to about 500,000 cells in total. So here's a picture of the reality. So because these call-in samples are quite weird and we're only interested in maybe specific regions, and it's very time intensive and expensive to image the entire tissue, we're instead selecting specific windows where we want to image the tissue. And those are these field of views. So we're not covering the entire tissue, but only selected regions. The features that we get are, on the one hand, morphological stainings, such as CD45, CD3, DAPI, etc. These are fluorescent values that we have in the end for every cell. And most importantly, we measure about 1,000 features covering cell type, cell state, cell-cell interaction, hormone activity. And just to say, this is a general panel set up by NanoString. It is not specific to this study, but to any study. But additionally, we could include 20 custom genes specific to our tissue and disease of interest. We start out with sort of a classical single cell analysis, because this is not Visium data or some other sequencing-based technology. We do in fact have single cell resolution after having segmented our cells. So we can do normal dimension reduction. Here are UMAPs of our 9 samples. And then we did a unsupervised clustering, and we saw that there's some spurious subpopulations that only show up in certain samples. So there seems to be quite a batch effect, whereas the healthy samples were quite similar to one another, which is very promising, as each of these takes about a week to image. Secondly, we integrated our samples, and then we find that the unsupervised clusterings gives more consistent profiles in the frequencies of subpopulations. And we find that our 3 healthy controls are very similar, and then the diseased ones are all quite a bit different, but some samples are more similar than others. And what we can do is to then just look at the same plot, but coloring here by condition. And we see that the healthy controls in the middle in blue, they lack cells in this area that's highlighted in a box, whereas the inflamed samples have a lot of cells there, which seems to indicate that that's sort of the cells that are distinguishing our diseased from our healthy samples. Also, if we do an embedding on the samples, so not on the cell level, but on the sample level, we get a plot like this. So on the bottom left, we see our 3 healthy samples, and there's 3 diseased samples that seem to be also on this side. So they're maybe not as inflamed, whereas the samples on the right seem to be quite distinct. So we could also confirm that these were indeed the most diseased patients. We then did a label transfer using a very big single cell reference data set to annotate our cells into broad subpopulations. We then grouped these into biologically meaningful compartments, namely epithelium, myeloids, plasma, stroma, and T cells. And within each of these compartments, we did again a label transfer to arrive at a much finer, higher resolution annotation, which we then worked with for downstream analyses. And this is again shown here on the left, upper UMAP, coloring by subset, on the right by the higher resolution. And on the bottom, I'm just showing the immunostaining. So for example, we can see that the epithelium is high in pan-CK and that sort of thing. Okay, at this point, I sent all of this to my collaborators in Spain, and they said, we think the annotation looks great. Could we see something in space? Because that's the point of doing spatial transcriptomics. So I sent them plots like this, which were easy to generate, but not very useful. They came back to me saying, it's really hard to tell apart the colors. Maybe we could get a plot per subpopulation. So I tried this and I ran into a lot of issues because I was generating interactive reports, interactive plots, and that was just a lot. And the reason for that being is we have 20 samples times 20 field of views times about 50 clusters, which gives a couple thousand plots to look at every individual subpopulation and every individual gene separately. So we couldn't really, I could generate this, but there's no way to look at this. So we spent a lot of time looking at different plots. And a week later, they selected very specific regions in the tissue that they were interested in, with which we then proceeded. And here are a few of these selected regions. So here, instead of coloring by cell type, we're just coloring by compartments. So we can really nicely see the plasma cell, the epithelium in red, giving these bubble structures or layered structures on the cell border. And we can then look at just one of these and color by subpopulations again. But to then see a specific cell type is still very hard because there are just too many colors and too many cells. So what we can then do is to sort of fade out all the background and highlight a specific cell type of interest. And here the cell type of interest is highlighted in yellow. So all of that is just to show that there was weeks and weeks of developing ways of visualizing the data to see what interesting we could find, because just showing all cells is really not that interesting. Even if we know there's an interesting subpopulations, it's hard to see. Just maybe one more example. So here we have the epithelium again. And instead of just coloring the cells in red, we can then find markers for that. So EpCAM, for example, AQP8, which is nicely expressed on the cell, on the tissue border, and similar kind of visualizations for other compartments here, the stroma and the myeloids. So in this way, we were really able to get a better understanding of our data by just playing a bit with the visualizations, the sizes of how to visualize cells, etc. And then maybe one more to maybe what I said was that they manually selected these regions, which is of course not feasible and not very scientific at all. So we should instead use some statistical methods to identify such regions in the tissue. And one very basic concept that's useful here is complete spatial randomness, which, without reading the definition here, just means that a signal is not at all associated with the spatial coordinates, that there's no spatial structure in the points that we're seeing, in this case, cells. If there is a structure like contagion, we would see clusters. Whereas if there's inhibition or competition, the cells would avoid each other and sort of give a very regular pattern like a grid. And that's shown here. So on the left, we have a regular pattern, on the right, a cluster pattern. And there is a lot of tests for these from the 70s that are really easy to run and fast and well established. One of these is the quadrant count test, which just counts the number of points in certain bins in a grid and sees if these are uniformly distributed or not. And by doing this, we can, for example, identify these 16 field of views for the epithelium. So that is just to show that for future analyses, we would want to avoid manual labor and instead use such tests to identify interesting regions in the tissue. And maybe one more example, we can also analyze how different cell types co-localize or avoid each other in the tissue. And for this, we simply use a correlation test on two-dimensional kernel density estimates of the cells between different cell types. And on the left is an example of avoidance. Unfortunately, there's very few cells here in red. On the red, contagion, where certain cell types tend to co-localize together within certain regions in the tissue. To summarize, we were really happy to see that COSMX does enable us to do cell typing, even though we're not at the whole transcriptome level yet. But we're hoping that by having a more targeted panel design in the future, so instead of 20 custom genes, maybe 200 custom genes, we will be able to do cell typing reference-free, so in a purely data-driven way without using single cell data. And secondly, there is a lot of data to look at here, because what I didn't mention is that this was all cell level, but in reality, we have molecule level. So these points, every point that we see has maybe 1,000 molecules in it, and it will be very challenging to visualize these. So we hope to use spatial statistics to identify what's interesting, for example, which regions in the tissue are interesting to visualize. Of course, analyses are always performed on everything, but for visualizations, we need to identify what's interesting and make choices there. And with that, I am done and happy to take any questions if we have time. I'm gonna unmute my computer. Yeah.
Thank you, Elena, for the very nice talk. And I think that, is one of the points that is clear that this new kind of data, is very rich. It brings a lot of information, but then we have to be able to exploit in a feasible way, somehow in an automatic way, even to identify. So it's something that, we are enthusiastic when we have this new kind of data, but when we have to think that, we have to deal with them. So I think that this kind of presentation is really pointing that we need, automatic method, bioinformatics method, so to avoid to have this, painful days and days of trying to visualize and have some automatic way to extract information. But aside from this consideration, so I don't know if there is a question in the chat. Let me check. Otherwise, I have a couple for you. So I think that trigger my mind when I saw one of your first slides, that one of the genes, one of the proteins that you can try to identify in the panel of genes are ligand or receptor. It's something related to cell communication, which is one of the topics. So I was just curious if and how you had the chance to work with that had even that genes inside the panel. And the second one, but I think is more probably a comment, is in the case of IBD, it's something that was proved to be very associated to the microbes that are present in the site. And in the last year, there's something called spatial metagenomics, so something like what you should hear, but for the microbes. I don't know if you have the chance to work even with that kind of data, so just a comment from you.
Okay. Regarding the first, there is a ligand receptor analysis. In fact, when we get the data from NanoString, because it's early access, they do a preliminary analysis and they send us these huge reports. But we did our own analysis, of course. But at this point, this maybe just wasn't the main interest. It was more to validate things that they had done experimentally. So there's lots of microscopy data, single cell data, where they have many more replicates, where they already do the analysis they want to do. And here the goal was to use this new technology to see if we could find these things in a data-driven way, just using spatial transcriptomics, but not finding new things. But can we find what we already know only using this data? So this was, I think, the main goal here. But yes, you can do ligand receptor analysis, cell communication. And regarding the second thing, I don't know. I don't have a lot of knowledge, to be honest, about the biology here. I'm mostly relying on my two amazing PhD collaborators to interpret the data. Yeah.
Great, great. Thank you. So I think, I don't know if there are other questions, but I think that we are a little bit out of time, so we can save them from the final Q&A, and or just to report to Elena if we have the chance to answer after her presentation. And I think that we can say thanks again to Elena and move to the next speaker. So I leave the stage to Enrica that can introduce the speaker.
Thank you, Giacomo. Good evening, everybody. We can now continue with the short talks selected from the call for submission. These talks do not have question time at the end. Anyway, feel free to put your question in the Q&A chat, because we will have if we will have time at the end, we will be more than happy to have them for the roundtable. So I leave the stage to Eva Viesi from the University of Verona. Please, Eva.
Thank you. So, good evening, everyone. I'm introducing you a new space-based method for the downstream analysis of spatial transcriptomics data. So, the main topic is spatial transcriptomics, which is a technique that allows to characterize gene expression within the tissue context. And unlike other sequencing techniques like bulk and single cell, spatial transcriptomics retains the spatial coordinates of spots and cells on a tissue section. When clustering spatial transcriptomics data, we have to deal with different challenging tasks like the limited interpretability of the results, and we would like also to understand how much the spatial information combined with the gene expression information could improve and affect clustering results. These are some state-of-the-art spatial transcriptomics tools. So in particular, Biospace and Giotto use the first Bayesian model and the other the hidden Markov random field model to combine both the physical position of the spots and their gene expression, while Spagisi and STLearn also add some morphological and histological features when clustering spatial transcriptomics data. All these tools use some complex models and require different parameters to be set. What we proposed is a new space-based method which combines spatial coordinates and the cell spot expression matrix. And what we introduce is a new parameter which is called space weight, which has been implemented both in a linear and a non-linear way to obtain the final distance matrix that we use for clustering. So the first approach, Stardust, implements a linear weight of space. We first perform a linear scaling of the spatial coordinates distance matrix, and then we weight it with a parameter which is defined by the user and that describes the weight given to the space. Whereas the second approach, Stardust implements a non-linear weight of the space. And what we do is to normalize the gene expression matrix in a range between 0 and 1, and then we use these values to weight the spatial coordinates distance matrix and sum the transcription matrix. In this case, the space weight changes dynamically according to the distribution of the gene expression values. What we do then is, starting from the final distance matrix, we compute the closeness, which is the distance of all the spots to all the other spots, where a spot is represented as a vector of distances. Then we use these distances to weight the edges in the graph that we will give an input to a clustering algorithm. In particular, we use the Louvain clustering, which is a graph-based method divided in two main phases. the modularity optimization, in which nodes are moved in order to optimize the modularity function, and then are aggregated into communities of similar nodes. As evaluation metrics, we use the cell stability score metric and the coefficient of variation. The first represents the tendency of a cell to remain clustered with the same group of spots when we induce a perturbation to the dataset, for example, by removing a percentage of elements, whereas the coefficient of variation is described as the ratio of the standard deviation to the mean of the cell stability score distribution. What we want to have is a high cell stability score and a low coefficient of variation. We also use the Moran's index, which is an index of spatial autocorrelation, to evaluate the biological coherence of our clusters. And the Moran index uses both the genes values and the genes locations to compute the spatial autocorrelation. And the idea is that if similar are localized, then the autocorrelation is high and positive. These are our clustering results. So we compute the cluster stability for 5 spatial transcriptomics datasets, in particular 2 human breast cancer sections, a human heart, a human lymph node, and the mouse kidney from the 10x Genomics platform. We would like to see how stable was a cell or spot in a cluster, where 0 means not stable and 1 means completely stable. What we observed is that space was able to increase the cell stability score with respect to not using space, and also was able to increase the number of stable spots in the clustering task. We also observed that our 2 proposed approaches, Stardust and Stardust Stars, show lower coefficient of variation values compared with the other state-of-the-art tools. We compute also the biological coherence of our clusters on a human breast cancer tissue section, and we observed that our cluster regions were coherent with a manually annotated section by pathologists. As next steps, we would like to introduce a new method to speed up clustering computation, in particular for high-dimensional datasets from, for example, the SlideSec and SecScope technologies, and to extend our algorithm to use different clustering methods with respect to the Louvain method. Thank you for the attention.
Thank you, Eva. Now is the time of Giulia Cesaro from the University of Padova. So, Giulia, the stage is yours.
Can you hear me?
Yes. And now we can see you. So you can share your presentation.
It's sharing. Are you able to see? Okay. Now? Thank you. Thank you for the stage. So, hi, everyone. Let me introduce the main topic of my talk and also of the second part of the workshop, which is the cellular communication. Cellular communication mainly occurs through a 2 signaling mechanism, an intercellular signaling, which is mediated by the interaction of 2 proteins, a ligand and a receptor, and occurring between pair of cells, so a sender and a receiver cell. Then there is an intracellular signaling, that's a signaling that happens inside the receiver cell through the activation of a signaling pathway, which can lead to the activation or inhibition of specific transcription factor that trigger the cellular response to that interaction, which is the regulation of certain target genes. The advent of single-cell RNA sequencing data offers the possibility to study cellular communication, since it enables the identification of different types of cell composing a tissue and quantifying at RNA level the actor of the communication mechanism, so for example, the ligand and the receptor. In this sense, we develop SGS-COM, which is a computational model that infer and quantifies cellular communication from this type of data. The main contribution of the work is the introduction for the first time of an intracellular score, which is a score that wants to quantify the evidence of ongoing intracellular signaling as effect of the communication. To compute this score, we combine two information: a stat information using graph topology-based algorithms, such as the one proposed by Nichnet, on intracellular signaling network constructed by public database of signaling pathway and transcription network. And then we combine with a dynamic information through the quantification of the cellular response based on the transcription profile of target genes. The second contribution of the work is the proposal of a new scoring scheme for the intercellular signaling, with the main goal of addressing some limitation of current methods. For example, our score avoided the need to fix a threshold for the gene expression level. During the computation, account also for the cell cluster size and data variability, and use a matrix that works in a finite homogeneous range, making the score easily interpretable and portable across dataset. We think that the combined use of the inter and intracellular score enables the prioritization of the most strong and reliable signals, which are the one with a high evidence of an ongoing ligand-receptor interaction, so a high intercellular score, but also a high intracellular score, meaning that there is a strong evidence that we are observing the cellular response to that interaction. Here we present an extension of this method that enables the analysis and inference of differential communication between experimental group at both intercellular and intracellular level. The identification of changing in the interaction, in the communication between experimental group might have an important aspect in understanding biological process that regulates, for example, a pathological state or an exposure to different treatment. To identify differential communication at intercellular level, that's at level of single ligand-receptor interaction and between pair of cluster, we first obtain a distribution of values of the intercellular score in the two conditions, and then we apply statistical tests to assess if the difference that we observe in the two distributions is statistically significant. Differential intracellular signaling is identified instead at the level of the downstream target genes. Watch so if there are changes in the final cellular response. In this case, we first identify differentially expressed target genes, and using a Fisher test, we compute a score which is somehow a measure of how much the difference between the two experimental groups. Here is an example of application of our tool. In this case, we applied SGS-COM to a public dataset of amyotrophic lateral sclerosis and pathological normal donors to identify differential interaction between 10 different cell clusters that involves neuron, glia, and vascular cells. Among the different visualization that our tools provides, the heat map and core diagram, these slides show the number of interaction that our tool was able to identify as differential at both inter and intracellular level. As we can see, the highest number of altered communication involves neurons, which might be associated to the disease or to neurodegeneration. But we can go in detail and see which are the interactions that are identified differential. So at the ligand receptor level, the heat map allows to appreciate the difference between the two experimental groups. So the disease group in orange and the control group in green. And we can compare the intercellular score in two conditions, which is encoded with the diameter of the circle, but also see at the same time the difference in the intracellular signaling encoded with the color map. Finally, let me spend a few minutes to talk about the future direction of this work. Given the impact of considering different resources, we would like to extend our repository of ligand receptor databases of human and mouse, integrated in new resources such as the new version of CellPhoneDB database. Then we tested our tool on 2 spatial transcriptomic datasets, but we would like to further validate using other spatial data, but also other complementary data modalities, such as the one proposed by Liana framework. And in long terms, we would like to extend our analysis in time, so inference cellular communication over time, increase the visualization tool also to handle this type of data, and at the same time keep the low computational burden of our tool. So thank you for the attention for the talk. And if someone would like to discuss face-to-face about the work on differential communication, we are present at the ECCB conference on a poster session of Monday.
Thank you, Giulia. And also thank you to Eva. And we can move on the schedule. So please, Gabriele, you can introduce the next speaker.
Okay, so the next speaker is going to be Robin Broways from Ghent University, and his talk is going to present us how to model cell communication when you have multiple samples and multiple conditions. So I'm really eager to learn how to tackle such a complex task. Robin, the stage is yours.
Yeah, thank you. Can you all see my screen? Okay, that's weird because I see my slides in full screen mode. Okay, I will share again. Better now? Okay, great. Yeah, thank you for the opportunity to present my work here, which will be about modeling cell-cell communication from multi-sample, multi-condition single-cell RNA-seq datasets. And I have to say that this work follows perfectly on the work that Giulia presented in the presentation before, because it will be about differential cell-cell communication and also combining the intercellular with the intracellular aspects. With a short introduction first. So as you all know, single-cell RNA-seq has been used quite extensively the last couple of years to study cell-cell interactions in health and disease states. And the majority of tools will predict ligand-receptor interactions between sender and receiver cell types. And that's based on using a database of known ligand-receptor pairs and combining this with the expression data of your interacting cells. And the most famous tools for this are SelfNDB and CellChat. Also in the Liana framework, it's included here. And I'm sure that about SelfNDB and Liana, you will hear still a lot today in the talks of Luz and Daniel. And the second type of approach will actually go one step further and also incorporate the intracellular aspects of cell-cell communication. And here we will combine expression data of the cells with prior knowledge on ligand-to-target signaling pathways. And one such tool is NICHNet, but also a tool presented by Giulia before fits in this framework. And I want to say here that the main difference that NICHNet does is prioritizing expressed ligand-receptor interactions based on enrichment of downstream target genes in a procedure that we call ligand activity, which is a similar metric as transcription factor activity, but then using ligands as regulators and not transcription factors. Important to perform this ligand activity prediction is that you have a good model of which target genes can be affected by which ligands. And for this, we integrated several public data sources about ligand-receptor interactions, signaling interactions, and gene regulatory interactions. We integrated these in one weighted integrated network. And based on this network and graph algorithms, we built a model of ligand target regulatory potential, which indicates the evidence, the prior knowledge that exists about the potential regulation of a target gene by a certain ligand. And then this model is used together with your data of interacting cells to prioritize potentially active ligand-receptor pairs. I want to stress here that these two approaches, the one focusing on the intracellular regulation and the ones focusing on the intercellular aspect, they are both complementary, and that you should be aware when interpreting the output that this can be quite different. So tools like SelfNDB and CellChat will provide you with a complete list of expressed and cell type specific ligand receptor pairs between the cell type pairs you're investigating, whereas NICHNet will more focus on ranking ligands that might be acting on a certain receiver cell type. This is the case for steady state analysis, but also when you perform differential cell-cell communication analysis. So if you compare two conditions, as mentioned by Julia in the previous presentation, the interpretation is also different. Like the first class of tools will generate a list of differentially expressed ligand receptor pairs, whereas NicheNet will provide a ranking of ligands, of which there is some potential of regulating the DE genes in the receiver cell type. So I think there is still a need for tools that prioritize ligand receptor interactions based on both expression, differential, and cell type specific and activity, and which is also done by the tool of Julia, but is something we will do here in multi-NicheNet as well. Second limitation that multi-NicheNet will address has to do with the multi-sample nature of the data sets that are typically used to compare different conditions. So these multi-sample data sets, as the name says, these data sets will profile several samples, for example, patients from a certain disease like COVID-19, and compare those with healthy patients. If you would apply the most used tools in a naive way, you would first pool all the cells of all samples together and then analyze cell-cell communication, and then analyze the differences between cell-cell communication between your groups of interest. But this pooling of cells is suboptimal from a biological and statistical perspective. From a biological perspective, you ignore then that cell-cell communication actually happens within one patient. And statistically spoken, you ignore that there is variation between samples that you should take into account. And moreover, it might be more challenging to handle batch effects, correct for covariates, and also differences in cell numbers between patients. So for these reasons, and also for the reason that the current tools in their default output, they don't provide much insight in interpatient heterogeneity, we think there is still a need for a novel tool that can handle these multi-sample challenges. And this, together with the need for prioritizing both on expression and activity, we developed multi-NicheNet, which will perform differential cell-cell communication specifically for multi-sample, multi-condition, single-cell or NiceSeq data sets. How does it work? It's based actually on the Muscat framework that was developed by Helena, also one of the previous speakers of today. So we start with the raw count matrix of our single-cell NiceSeq data. And next to that, we also need some metadata about the cells that indicate the sample of origin of cells, the condition, and a cell type annotation label. This information is then used to generate pseudobulk count matrices for each cell type. So we will aggregate all the counts of all cells of one certain cell type per sample. And then in the end, we end up with one pseudobulk count matrix per cell type. And these pseudobulk count matrices can then be analyzed with classic bulk RD-seq differential expression tools like HR. We will use the output of this to determine the level of differential expression of the ligand, of the receptor. It also allows us to define a set of DE genes in the receiver cell type based on which we can then calculate NicheNet ligand activities. And we also use this information to get a measure of cell type specificity of both the ligand and receptor. Once we have calculated these factors, we will scale each of them and aggregate them in one final prioritization score. And this prioritization score is then used to rank ligand receptor pairs based on condition specificity. I want to stress here that if you have information about batches or covariates that you want to correct for, you can easily add them to the metadata, and the model will account for those in the prioritization downstream. So in the second part of my presentation, I will go over the type of visualizations and results that multi-NicheNet can generate on two case studies. In the first one, we will look at tumor microenvironment interactions in breast cancer that are linked to response to NTB-W04 immunotherapy. So for this, we used publicly available data from the Dieter Lambrechts lab. So these researchers generated single-cell RNA-seq data of breast cancer patients before NTB-W04 therapy and during NTB-W04 therapy. And they also measured the response to the therapy for each of the patients, and they could define two patient groups, one group of patients that show clonal type expansion and a group of patients that does not show expansion or only a limited amount. And then we can use multi-NicheNet for a simple comparison between these two groups of patients, and we will focus here on the differences that already existed between these two groups of patients before therapy. One of the first type of outputs that multi-nichenet can generate is these type of circular visualizations that show the top differential interactions between the two groups. This for all cell type pairs you want to analyze. But I want to stress here, okay, this gives a general visualization, but we also want to dig deeper into certain cell type combinations, into the data behind the predictions. So that's why we also generated several other types of visualizations, which I will go over now. So in the first type of visualization, you zoom in on a certain sender-receiver pair of interest, in this case, macrophages and CD4 T regulatory cells. And what we will do then is visualize the expression of each prioritized ligand receptor pair per patient. And we also highlight whether a certain, whether there were enough cells of both the sender and the receiver present in order to perform a proper differential expression analysis or not, which can be useful information for the downstream interpretation. This shows the expression. Because we also prioritize based on nichenet activities, we will show the nichenet ligand activities next to this, and we split this up in both upregulatory and downregulatory activities. And this is the kind of main visualization output you get from multi-nichenet. You can extend this and also look at other cell types. So we will here look at CD4 T cells and CD8 T cells as receivers of macrophages. And one thing that we can clearly see here is that many PD-1 signaling interactions are more highly expressed in the group of patients that will show clonotype expansion after the therapy. And this is a logical result because the anti-PD-1 therapy will only have an effect if PD-1 itself was an important molecule in the tumor microenvironment before applying the therapy. I want to stress here that these PD-1 signaling interactions are not only differentially expressed between the conditions, but you also see a higher activity of PD-L1 signaling in the patients that show expansion. So this means that there are some target genes enriched downstream of PD-L1 in these patients. And in the next type of visualization, we can look at the specific target genes. So in this plot at the left, you see the ligand receptor pairs. At the top, you see the predicted target genes. This is done by the nichenet framework. But in addition to that, we can now also exploit the multi-sample nature of the data to filter and only keep ligand receptor target links that are supported by expression correlation, because we expect some correlation expression between the ligand receptor pair across patients and the target gene across patient. And we typically see that an outlier patient in a group at the ligand receptor level is also an outlier at the target gene level. These target genes are predicted by prior knowledge. So you can also have a look at which specific signaling interactions downstream are likely to be active here. And for each of these interactions, you can also look at the specific data sources that we integrated and that support each of these intracellular interactions here. And I want to say that many of these interactions are now supported by data sources that we added to the updated version of the nichenet networks, which is the nichenet v2 networks. Okay, a last thing about these visualizations here is that some target genes downstream of ligand receptor pairs encode for ligands or receptors themselves. So these might then also affect other cell types. And this made us think that we can start to look at intercellular regulatory networks to maybe infer signaling feedback and cascade mechanisms. Like shown on this diagram, PD-L1 is a downstream target gene of the interferon gamma interaction, and PD-L1 itself is involved in an interaction towards CD4 T cells in some kind of feedback mechanism, but it's also involved in interacting then with other cell types like CD8 T cells. And with multi-nichenet, you can generate these types of intercellular regulatory networks automatically and compare between the groups you're interested in. So here we compared the groups of patients with expansion versus the group of patients without. And we see many more of these intercellular signaling links in the group that show expansion after therapy. Okay. Last thing about this case study is that we can also tackle some more complex questions and perform comparisons between multiple groups and exploit data sets with more complex experimental designs. So in this data set, we had single-cell RNA-seq data before and during therapy. So we can now ask the question, what are the intercellular signaling changes that are induced by the therapy, and how are these changes different between the two groups that we are looking at? And as we look at the results here, we see, as we can expect, that in the group of clonotype expansion, we see an increase in pro-inflammatory interactions like interferon gamma and interactions involved in antigen presentation. What was also of interest is that also in the group of patients that did not show T-cell clonotype expansion, there were apparently some interactions that still increased in expression. And they mainly went towards endothelial cells. And we zoomed in on those, and we found that indeed pathways like Notch and IGF-2 were increased here. But when we looked in more detail, we saw that this was only true for a subset of patients. So the majority of patients showed this increase, but many other patients in that group did not. And it's only because of the visualizations that take into account the intersample heterogeneity that we can kind of find these within group differences in cell-cell communication. My last slides will be about a second case study where I will highlight how you can use multi-niche-net to address cell-cell communication analysis on integrated atlas data. And here we showcase its ability to correct for batch effects. More specifically, we applied it on integrated atlas data based on 4 individual single-cell RNA-seq data sets that profiled healthy and lungs from idiopathic pulmonary fibrosis patients. After integration in one lung atlas, we can apply multi-niche-net and correct for the batch effects that exist between these 4 methods. And this is then, again, the type of output you can get from multi-niche-net with the top interactions that seem to be specific for idiopathic pulmonary fibrosis. I will not go into detail about this here, but I will only still say that some interactions are, of course, more highly ranked after batch effect correction than without batch effect correction. And I show a subset of these here at the left. In the visualizations, we actually show the corrected pseudobulk expression values. And you can see, okay, indeed, these interactions seem to be differentially expressed between the 2 groups we are comparing. In the next visualization, we show the non-corrected expression values, and then you see that this differential expression is less apparent because some data sets show actually higher expression of these ligand receptor pairs than others. But with multi-niche-net, we can correct for this. So to conclude, we developed multi-niche-net, which is an extension of niche-net, to better handle differential cell-cell communication in a multi-sample context. And it overcomes some of the limitations of current tools because it can now consider both expression and activity at once to prioritize ligand receptor pairs, while also taking into account interpatient heterogeneity, address complex questions, correct for batch effects, and providing visualizations to explore the data behind the top predictions so users can get more informed about these top predictions and use this information to go for further experimental validation. I would like to end by showing the links to the software, so you can find both niche-net and multi-niche-net on the GitHub page of our lab. I would like to thank my supervisor and the other peoples in my lab, and also the external collaborators from the University Hospital and the KU Leuven. Just a last note, our lab is hiring. We have still a lot of money to hire postdocs and PhD students for 4 years on the modeling of cell-cell communication in collaboration with many other scientists involved in bioinformatics, but also tumor microenvironment and liver tissue biology. And Ghent is, of course, a very nice city to live in, so that's why I make a bit publicity of this. So thank you for your attention.
Okay, thank you, Robin, for this very nice presentation. I have just a curiosity on the analysis that you showed us about within group differences in cell-cell communication. I was wondering, other than looking at this really nice plot, whether you can suggest or whether you're already implemented some statistical test to rank these differences, so to filter out, say, the top 10 most significant differences in this list of results that you get.
So you mean some kind of approach to find the patients and to discriminate the patients that show an increase versus the ones who don't within that group?
Exactly.
Yeah, we did something quite simple at the moment. And we just looked at the expression values of each ligand receptor pair. We get that for each sample. And then we can look at the differences before and during the treatment, and then rank based on this difference. And then we could get to 2 subgroups, actually in this case 3 subgroups, one group that showed a decrease, one group for which expression levels stayed the same, and then one group for which we saw an increase. We went deeper into the biology behind this because it could potentially be interesting, but it was not linked to breast cancer type, it was not linked to T-cell levels, it was not linked to like hot versus cold tumor architecture. So at this moment, we could not find any clinical difference between the 2 groups that we saw. But we looked deeper into the single cell data, and then we found some interesting differences in angiogenesis and lymphangiogenicis pathways between the 2.
Okay. Thank you again, Robin. In the interest of time, since we're already a little bit late, I'm going to leave the stage again to Enrica for the next speaker.
Thank you, Gabriele. So now it's the time of Luz Garcia Alonso from the Wellcome Sanger Institute. So Luz, you can start your talk. Thank you.
Hello, everyone. Thank you for staying until this long. So I'm Ruth. I'm coming from Rosert Vento Laboratory, which is one of the main authors of the seminal publication of self-on-DB. I think some of you might know it's a tool to uncover cell-cell communication for single-cell transcriptomics, and this was before the spatial transcriptomics era. So our labs shares interest with most of the people in this workshop in that we aim to understand the influence of the microenvironment on the individual cellular identities. And here we have a big focus on the female reproductive system, where there exists an important knowledge gap if you compare it to other organs with diseases with similar prevalence. And basically, our research has three steps. So profile, predict, and perturb, meaning that as part of human cell atlas, we provide hundreds of human samples a year with single-cell technologies. Then we use computational tools to define the hypotheses. And then the third point is that we try to validate them using our biobank of organoids that we perturb and track with, again, single-cell genomics, imaging, lineage tracing, etc. And for this, we use and develop self-on-DB, which we have been adapting through our publications to the challenges of our own datasets. And in my last slides, I will give you an example of this whole process for us. So I think the field of modeling cell-cell communication is quite broad, actually. So it comprises a plethora of different applications, and there are excellent reviews out there, such as the one from America Mengoli in Nature Genetics last year, from Almet and other cell-chat developers, and also from Daniel from Self Rodriguez Group that did a really nice comparison of methods and databases, and I think we'll be presenting later. So generally, the main applications of cell-cell communication modeling can be grouped into 3 areas. So the first one will be understanding cell differentiation of development, when you are most likely interested on the dynamics of a specific cell type or a cell lineage, and you want to uncover what are the cell extrinsic factors that are pushing this differentiation forward. Another scenario will be that you are just characterizing the whole tissue or organ's homeostasis, and for example, you just want to understand how healthy tissue. And another application will be when you want to study the full microenvironment, such as the immune compartment, most likely in the context of disease, such as cancer or inflammatory diseases, and need to characterize how the interacting partners change or evolve globally in the microenvironment. Here we're not talking about specific signals on a specific lineage, but more about combination of signals in a more broader cellular composition. So now, depending on your research question and the input data you have spatial or not, you will require a specific methodology tailored to your hypothesis. And it is quite unlikely that a method will be able to answer them all. So, this might explain why the predictions of different methods just do not correlate, because they aim to answer different questions. So we see that modeling cells and communication has 2 big components. One is the method of the approach that you use to interrogate your data. And as I said before, it depends on your research question. And then the other big block is the prior knowledge or the database of ligand receptor addition molecules that you use to infer cell-cell interactions. And this is how we like to structure CellPhoneDB. So CellPhoneDB has a battery of methods to interrogate your data. For example, in the original version 1 and 2, the methods were tailored to interrogate full datasets, all the cell types in the tissue. And more recently, we released version 3, where we added a novel approach that allows the researcher to focus on one lineage and is cross-talk with the remaining cells, and is a more tailored study when you toward more analyzing the differentiation, the dynamics of specific cell types. So now going back to the triangle, with this approach, we can answer more specific questions, such as the molecules that are making the transition in between two cell types. For example, this helps us to identify what we need to add in the in vitro media to convert the cell A in another cell type. So with the latest version of CellPhoneDB, we also allow the user to include spatial information. And we have been brainstorming in our lab what's the best way to use spatial transcriptomics, such as Visium, for modeling cell-cell communication in general atlases. And we always get stuck with the same bottleneck, that's the specificity of the underlying ligand receptors. So we have a tendency to think that interacting ligand receptors should coexist or co-appear in the space. So you should expect some kind of spatial correlation. But how do you compute this correlation when a receptor can interact with several ligands, or a ligand can interact with several receptors? And this is what happens with most of the signaling families as TGF-beta, NORCH, or WNT. And I leave this as an open question for further discussion if you wish. So because of this, at the moment, the way we use spatial transcriptomics is to locate cells and discard interactions that cannot occur just because the cells do not co-locate. So in CellPhoneDB, we have our own database of ligand receptors and addition molecules that we create manually. So we believe this is key, as the reliability of your predictions will depend on how good is your database. So for this purpose, we are active curators of this type of data, meaning that all the interactions that we'll include in CellPhoneDB as curated are actually manually reviewed by us by reading the supporting literature. And you will appreciate this is a lot of work, and to go through all the papers. So basically, there are 6 essential factors that we consider to be important in a database to model cell-cell interactions. So the first one that I would like to highlight very strongly is the inclusion of protein complexes. So far, I think it's only CellPhoneDB and CellChat that are accounting for this key element. So you know that in general biology and cell-cell communication specifically, so most of the proteins work as heterodimers, meaning that the signal will only be received and transduced if you have all the elements of the complex. So in some cases, depending on the composition of the heterodimer, the response will be different. And here I'm putting the example of the TGF-beta receptors, that depending on which are the modules assembling as receptor, you will be activating a different SMAD transcription factor. So if you do not model this accurately, meaning that the sender molecule or the receiver is not containing all the elements, so you might end up predicting an interaction that's not true because you lack one element. And this is basically a false positive. Now, if you have many of these in your predictions, you will have many false positives. And I think this is a risk you don't want to take into account, you don't want to take if you are going to spend a lot of money validating the predictions. Another aspect is that not all the proteins that have been observed to physically bind in vitro have a function in cell-cell communication in vivo. So now there exists lots of methods to measure in a high throughput way protein binding, but the fact that two proteins can stick does not mean that the binding is functional and we have a response. So this is why it's good to separate this high throughput screening from protein interactions that have an actual functional evidence. And the third point is that now in SelfOnDB, we are starting to include ligands that are non-proteins. And this is because there are other molecules other than proteins that can act as ligands. Some examples are small molecules such as histamine or steroid hormones such as estrogen and progesterone, which are key for us in the reproductive system. And to do so, how are we including this data in SelfOn? So we basically are reconstructing the biosynthetic pathways and using the last representative enzyme as a proxy of abundance of the ligand. And as I said, this is already available in SelfOnDB4. And now we are in the process of adding novel layers. One is trying to connect the receptors with the downstream transcriptional effectors. This is important because it gives you further level of evidence that the ligand receptor binding is happening. So if you have information on the downstream status of the transcription factor and you know which transcription factors are active in your data and you know which receptor can activate transcription factor, then you can prioritize the functional ligand receptor bindings. This is an idea that's been explored by Nitzner, just presented before, but here we propose a much simpler approach where we only focus on receptor and transcription factor pairs where there is a direct evidence that there is high specificity in the interaction. So you cannot doubt that whenever the receptor is activated, the transcription factor will be activated. And here again, I'm going back to the example of the TGF-beta receptors activating small transcription factors, or even more specifically, nuclear receptors that are at the same time receptors and transcription factors. So we are also in the process of including a major release with neurotransmitters, which coupled protein receptors, and this with James and Kazumasa from Sarah Teichmann team. And I think this is very exciting because this family of interaction is quite underrepresented in all the databases so far. And I think it's key for modeling or studying the brain nervous system. And actually, most of the benchmarks on spatial transcriptomics are done on the 10x dataset on the brain. So I think this will be quite useful. And finally, I'll also just mention, I forgot that we are also annotating the directionality of the interaction. So who's acting as sender, who's acting as receiver. And I think we think this is key if you want to link the data with single cell ataxic data. And just to conclude, I'll show you an example on the use of SelfOnDB in our lab. So here in this study, we are atlasing the human endometrium during reproductive age, and you might not be interested on the biology, but I'm trying to convince you that this is a very nice data set to benchmark your tools and your methods. So here we are in front of a tissue that regenerates only, and this regeneration has 3 steps that's coordinated by the hypothalamus pituitary axis. And basically you have first stage with menstruation, but basically you shed the whole endometrium, so the endometrium disappears. Then we have another stage that's proliferative stage, where basically the tissue is repaired and it proliferates. And after ovulation and secretion of progesterone, the tissue differentiates and desidualizes to get ready to welcome an embryo. So here we perform single cell analysis and we observe cellular heterogeneity larger than expected in the epithelial lineage. And when we look at the location of the cell states with Visium, here we were using cell2 location. So this revealed that each state, each cell type was located in a different layer of the endometrium. And the question here is, how is this diversification of epithelial cells achieved? What are the signals sent by the stroma cells or other cells there that are making this patterning of epithelial cells in the endometrium? And to answer this question, we had to release SelfOnDB3, where you can do a hierarchical analysis. Here we did it within the epithelial lineage to uncover which are the ligands and receptors that are specific to each epithelial state that I just described. Then we identify what are the other cell types sharing the microenvironment with those epithelial states. That is, what are the cells that could localize and could be sending or receiving the information from those epithelial cells. And then this basically allows us to prioritize high confidence interactions. And just make long story short, by ranking the prioritized interactions, we could identify that a potent inhibitor of Wnt, that's called DDK1, was secreted by the stromal cells in the functional layer. That's in the middle of the endometrium, where the epithelial cells differentiate into glands. So this drives us to suggest a model when we have a balance of Wnt and Notch signaling, and this balance is basically different in the lumen or in the functional layer, where the epithelial cells become glands. And so basically the question now is this model right? Is it true that you have high Wnt, low Notch environment in the surface and a high Notch, low Wnt environment in the lumen? And to answer this question, so we designed an experiment where we culture epithelial endometrial organoids. We had the controls, and then also we added either Wnt inhibitors or Notch inhibitors to simulate this balance. And the experiment showed that indeed, where we were inhibiting Wnt, we were able to generate more cells that look like granular cells. And instead, we were doing the opposite, where we're blocking Notch. We were instead obtaining more ciliated cells that are characteristic of the surface of the endometrium. And in the interest of time, I'll just conclude my talk here. Thank you so much for listening. Please get in touch if you wish to use any of the data sets that I presented to benchmark your methods. They are all publicly available. We also have other in ovaries and prenatal gonads. And basically here in this slide, you have all the people involved directly or indirectly with SelfOnDB and the study, and happy to take any questions. Thank you.
Thank you, Luz. Are there some questions from audience? Maybe I can start with mine. It was a really, really interesting talk. I just wondering if, do you consider only the intracellular signal that start from a receptor to a transcription factor, or you can also analyze those intracellular signaling that do not end to transcription factor? I was thinking, for example, to all those signals that end at an ion channel in the mitochondria, for example, that can also trigger different phenotype of the cell. And then I have also another question. I really appreciate that you can consider protein complex at the receptor level, but also transcription factor can act additionally each other, and also sometimes are mutually exclusive, or one silence another transcription factor. Can these systems be represented in your tool?
Okay, regarding the first question, so intracellular signaling, we are not considering this so far. I think it's quite challenging because you have a massive amplification. Most of the receptors do not have a specific or do not activate directly a transcription factor, but they need to go through a complex system of pathways. And I think it's really challenging to propagate the signal in those scenarios. So that's why instead of that, we think, let's just focus on receptors that you know that they are gonna specifically activate one transcription factor because it's been validated, and there is no way that this receptor is gonna activate other things because it's been validated. Of course, this restricts the scope of the transcription factors you can model, right? That's why you need other methods like the internet. But so far, we are stopping here, just only focusing on those. And the other question was on the fact that transcription factors can also work as heterodimers, right? So, okay, I think this is completely another field, and it's true, we should consider that. And I guess, again, the challenge here is trying to understand which is the target of each transcription factor and how targets change as you change the heterodimeric composition. And I think so far this is not, I mean, the coverage of knowledge we have for transcription factors working in this way, it's really limited as well. Even for transcription factors working as single proteins or homodimers, we don't know which are actually the targets, right? We can use. Now it's very exciting. It's a lot of things we can do.
Biology is always more complex than our models, so that's the problem.
But on the cell-cell communication side, I think it's quite striking because there are lots of receptors. We know they work as heterodimers. We know the composition, and you really need both elements. So if this knowledge is there, I think we should use it. Yeah, you don't want this false positive ending up in your results.
And also, if I can give another comments to the topic, I think that this field will gain also more information from the integration from multiomics, because signaling pathway is an integration between proteins, RNA compounds. So probably multiomics, maybe in single cell, could be one of the challenges in the future. So we can move on with the schedule if there are no questions, but there isn't. So the next chair is Giacomo. So Giacomo, please.
Yeah, thank you, Enrica. And I'm very happy to introduce the next speaker, Daniel, from the University of Heidelberg, that is going to talk about the role of the bioinformatics method that we use to do our cell communication analysis. How important is choose the right, let's say, method and the right database, or to better say what it means using one method or one database compared to another. So this is the topic of Daniel talk. So please, Daniel, the stage is yours.
Yeah, well, I hope you can hear me. And yeah, so yes, yeah, my name is Daniel Dimitrov. I'm from the group of Julio César Rodriguez, and yeah, I will present briefly, as Giacomo said, on the topic of cell-cell communication, inference from single-cell RNA-seq data, and it's a comparison of methods and resources. But for the sake of time, I'll basically call it Liana's manuscript. Yeah, I think we already had quite a nice introduction of basically why we want to predict cell-cell communication from the previous speakers, and this has been fueled from the recent technological advances in single-cell spatial data, and it's very exciting because it's we see that it plays an important role in homeostasis development in disease, and having the ability to make such inference allows us to really make like an additional layer of insights to kind of obtain an additional layer of insights. So I think this slide is now more or less clear. I will just very briefly go over it. But so Liana's manuscript focused on mainly on ligand receptor cell-cell communication methods, in which we kind of make predictions between different pairs of cell types or cell identities or clusters, and we use the mean ligand expression and the mean receptor expression. And this is the most common case, right? We use the ligand receptor notation, but often it doesn't necessarily include secreted ligands and membrane-bound receptors, and this is more or less the case for basically all tools that have been developed in this context. So this does not capture tools such as NicheNet or tools which focus on downstream signaling, and instead we see those as complementary, as also Robin mentioned earlier. And yeah, so what is often the case is that each tool, so ligand receptor tool, comes with a prior knowledge resource about the interactions that are basically possible to be inferred. And as Luz mentioned, I also believe that this is like kind of a very important part of the data set, but unfortunately of the predictions. But unfortunately, very often we see that the method and the resource come in a fixed manner, right? So it was more or less unclear what was the impact that the choice of a method and a resource can have on predictions. And this was more or less the motivation to build Liana and to use it to assess basically the impact of the choices that we make when we predict ligand receptor interactions. And yeah, as I mentioned, interaction knowledge is essential. So one of the main focuses on Liana's manuscript was actually to look in depth into the differences between the resources. And yeah, I think in this presentation I have presented only a small portion of this the results that we found about the resources, but I think they're also quite important. So the first thing that we noticed is that we basically made a pairwise comparison between all resources that we have in Liana, and this is 16 resources, each of which is ligand receptor based, and this includes cell phone DB, cell chat, and all the other really valuable efforts that have been made prior to Liana. And what we saw is that very often, even though the ligand receptor resources come from similar original sources, and many of which are actually manually curated, we see that there is some variability in the overlap between the resources. Or in other words, while few interactions were unique to a single resource, the overlap was kind of variable, right? So this kind of prompted us to look into the question of do this variable overlap also propagate into the potential functional insights that we can obtain when we use different resources, right? So what we did was that we used well-known biological databases to look for any functional biases in the resources, and this is done relative to the combined knowledge of resources. And this is what this plot represents. It's simply it shows a relative enrichment of interactions matched to pathways from signaling, which is a general knowledge of basically these 14 pathways. And we did this enrichment for each of the resources. And what we see is that indeed there are some differences in well-known pathways such as Hedgehog and Wind. So one could assume that these differences also would propagate in downstream inference, right? But so in the manuscript we also do this a bit further with more general knowledge databases. But for the sake of time, I will just present this one as a reminder that any prior knowledge comes with its own biases, and yeah, we should keep that in mind. So since the main goal of cell-cell communication methods is inference, we also wanted to see if using different methods and different resources would actually affect the inference, right? So what we did is, as a next step, is that we explored the relative agreements in the cell-cell communication predictions when using different methods and resources, quite simply, right? So what we do is that we use Liana to run all combinations of the 16 resources and 7 methods, so each method resource combination, and then we calculate the agreement between each of those runs, basically. And what this shows is, I guess, a bit overcomplicated graph maybe, is that it shows a figure. What it shows is the pairwise Jacquard index in the top 1,000 interactions predicted by each combination of method resource across 6 data sets. And here the blue line represents the overlap between the methods, and the red line represents the overlap between when using different resources. So what this really shows us is that basically both the methods and the resource could potentially result in limited overlap. Of course, this comes with some limitations, but like this type of oversimplified analysis, but it's something that it should be bear in mind. And this is what also prompts us to look a bit further into how the methods actually perform, right? Because we more or less anticipated to see such a results for the resources given their variable overlap, but this was not necessarily the case of what we expected to see for the methods, as many of them, at least in this scenario where we look at steady state data sets, we assume that they might be a bit more, they might agree a bit more. So as a first step to indirectly evaluate the methods, right? And I say indirectly because I want to stress that it's challenging to evaluate cell-cell communication inference because of the complexity of cell-cell communication. I think many of the previous speakers touched on these ideas, but it's very complicated it's challenging to obtain a ground truth that actually captures the complexity of cell-cell communication events, right? So instead that's why we indirectly evaluate, right? So the first thing was to check for the robustness of the methods, and we did this by introducing noise. And for example, we introduced noise in the data by basically what is presented here are the true positive rate of predictions obtained with different proportions of reshuffled cell labels. So what we do is that we reshuffle cell labels with different percentages of cell labels, and we start from basically 5% and we reach 40, right? And what we saw was that more or less most methods were fairly robust, as I would say that 40% is quite a significant proportion of cells to be reshuffled. And we see also that cell phone DB, cell chat, and single cell R, for example, were a bit more robust maybe than the remainder of the methods. And we also did a similar analysis where we assessed the robustness of noise to the resource. And again, we saw that the methods were fairly robust. So, which you know is quite comforting given the like limited overlap that we saw in terms of their highest predicted interactions. So as a next step, what we also wanted to do is to check if the ability of the methods to actually detect biological signal. And we did this by using what we call alternative code, at least alternative modalities. And in the first example, this is represented by the agreement with cytokine activities, right? So what we do is that we estimated cytokine activities in target cell types using cytokine signature from cytosig together with the enrichment tool developed within our lab called decoupler. And we used those active cell types, active cytokines within the cell types to kind of look for the agreement between the cell-cell communication predictions and the cytokines inferred to be active. So what this plot here represents is the odds ratios of interactions with matching active cytokines in the target cell types. And how you can interpret this is that basically a higher the odds ratio, the higher the agreement for a given range of ranks. And what we saw here is that most methods do agree with inferred cytokine activities, as we see a clear relationship between the inferred cell-cell communication ranking and the active cytokines, or in other words, we generally observed an enrichment of active cytokines in highly ranked interactions, right? So that was quite comforting. And similar to this, we also saw that methods also agreed with protein abundance when we used site-seq data. Another indirect way to see whether the methods actually capture biological signal was to leverage spatial information under the assumption that spatially adjacent cell populations are expected to have a higher chance to interact with each other than those cell types which are more spatially distant, right, physically distant. We did this by using spatial mapping from 8-10x Visium slides to identify colocalized cell types within the tissues, and we do this using Spotlight, basically using single cell matching single cell data sets, and then we run Liana on the single cell data sets. And again, we in a similar type of plot as the previous one, we see this relationship between prioritized ranking and spatial colocalization, which is again a comforting result. And this is in particularly a bit more visible in brain cortex when compared to, for example, breast cancer, which is likely due to the kind of more well-structured brain tissue. And yeah, so just to conclude, Liana is a ligand receptor analysis framework, which is easy to use. It provides a predictions consensus. It works with multiple data set, with multiple resources, and it's available on GitHub. But what I think is the more important message of these talks to build on, and to also make sense, build on what was also shown, is that methods and resources can impact the cell-cell communication predictions. And even though cell-cell communication methods do detect biological signal and are concordant with the alternative modalities, there is certain limitations and assumptions that come with those methods. And I think what is quite nice also from the previous talks, just to make it a bit shorter, is that many of those limitations and assumptions are now being addressed. And yeah, I think this is also where the direction of Liana is going. So we're interested in looking into multi-context insights, so moving beyond steady state interaction predictions, using either matrix factorization techniques or for example Muscat, as Robin showed, and also as basically all the three previous talks showed, in the integration of downstream signaling in spatial information. Even though here we used it as a kind of an indirect evaluation, is really something that can be used to support ligand receptor interactions. And this is more or less where I will end my talk. And yeah, thank all my collaborators and my lab. And yeah, it's more or less it. So thanks for your attention. I will now stop sharing.
So thank you, Daniel, for the very nice talk. And I mean, I would say that, you know, I feel the same sense of, I would say, frustration when we need somehow to prove that when we try to publish that our score is good, is reasonable, is somehow meaningful to be published. But we have the challenge of how can I prove? I mean, each score is different because we are measuring, we are modeling the communication different way. We have no ground truth. So I mean, it's not a trivial task, let's say. So, and I mean, I think that the Liana framework is one of the best ways to highlight this point in bioinformatics on cell-side communication. And speaking about the fact that different scores are modeling different things, and the impact of the resources in terms of legal set database, as you proved in your publication. So, you maybe, in your opinion, move our attention in identifying, let's say, some scenarios, so some cluster, some type of score, some type of databases that are maybe better fit a kind of biological analysis, because otherwise, really, I don't know. I mean, I think that is the question, which is the database that I should use and which is the method. So maybe, I don't know, I know I'm asking you the question to be, I mean, just to, you know, your opinion on this.
Yeah, I think it's, as you mentioned, it's really challenging. And I hope one can note is that I really refrain from actually suggesting that our benchmarks actually can tell which method performs best, right? So I really try to avoid this as best as I can. And what I can only say is that there is signal, but which method performs best, I'm not sure. And which resource is best, I think it's also, there is like a trade-off between coverage and quality, as always. So in a current version of Liana, we try to kind of do a balance between those, where we include all literature curated interactions, but what we saw is that also what is termed literature curation or manual curation also differs a lot between the different resources. So it's really challenging to say which one is really best and which one is not, right? So I would really not comment.
No, I mean, it's something that we should tell to the reviewer when he asks you to do.
Yeah.
Okay, do some comparison with the other score. I can do a Venn diagram, but eventually just something that maybe is not what you have in your mind. So I'm happy that we have the same feeling about this.
Yeah, I agree.
Okay, so let's see if there are questions from the chat. Otherwise, I think we can go on and save some of them for the last Q&A session. So I don't see any of them for now. So I will say thanks to Daniel.
Thanks for having me.
Yeah, okay, so maybe I can take the stage and introduce to you the next speaker. But before actually I do that, I would like to remind you that for the next two talks, we won't be taking live questions, but please, if you have some, type them in the live Q&As so that we can address those all at the end of both talks. So let me introduce Alice Chiodi from the Italian National Research Council. She will be presenting Ulisse, a package for going beyond simple gene sets when you are studying cell-cell communication. Alice, the stage is yours.
Can you see the screen? Okay, thank you. So I'm Alice, and I would like to talk to you about our R package, Ulisse. We developed Ulisse to study the interaction between gene sets, and it has two main applications. One is when the gene sets are pathways, so Ulisse is used to study the pathway, and the other in single cell data when the gene sets derive from different cluster or cell types, and Ulisse is used to study this cell communication. We will talk only about this part of Ulisse today. Ulisse needs mainly two input, a biological network and a gene list that can be or not ranked. We develop Ulisse to be highly flexible, so the user should have to provide its own biological network and have to choose if to build the gene list by using the expression data or the results of the differential expression analysis, as this data give different and it's up to the user and the study to decide which one to choose. Ulisse uses the input to calculate a score for each intercellular communication. So its main focus is on the cell type at the cluster level. We provide the information about the gene-gene interaction that build the communication, but our focus is on the cell type or the clusters. We provide also a statistical evaluation of the communication that can be used to filter the results to maintain only the significant one to build a intercellular communication network. The score is calculated. See here, we use the information of the genes of the different cluster to calculate the score, together with the ranks that are weights that derive or from the expression data or differential expression results. If we do not provide the weights, the score is equal to the number of links between the genes of the cluster considered, and it's this type of score that is used for the statistical evaluation. We calculated the p-value by using a permutation approach. In each permutation, we perform a random sampling to build two gene sets that has the same dimension of the markers, the gene set of the cluster considered. Then we calculated the score without the weights between these permutated the gene set and then use the value to calculate the p-value. We also calculate an empirical FDR score that is a corrected p-value that consider the permutation, and also provide a corrected p-value by using Benjamin-Hochberg method. We decided to evaluate our method by comparing it to other two packages. One is a single-cell signal array, that is a package that uses expression data together with a built-in ligand-receptor interaction database to calculate a score for each ligand-receptor interaction. So its focus is on gene level. It can use differential expressed genes results to identify gene-gene interactions that are specific of a cluster pair. Also, you can see here the score calculation. The author commented that the score can be used to filter the results by defining a threshold to remove false positive. They commented about the threshold, but it's completely up to the user, the choices of the threshold. The other package is iCellNet that uses expression data together with a database that is provided by the author to calculate an intercellular directed communication score. And also, you can see here the equation of the score. They do not provide any evaluation method to remove or reduce the false positive. We decided to test this package together with the two other one on 7 samples, 4 normal breast organoids and 3 breast cancer samples. We pre-processed the data by using Sera, performed the communication analysis and compared the results by using Spearman correlation, Jacquard, and structural similarity. So for each sample, we performed the communication analysis, compare the results by using Spearman correlation, to filter the results by using our statistical evaluation or threshold or the specificity information in this case, to then obtain communication network and compare the network by using Jacquard similarity, that is a measure of how many links of the network are shared, and structural similarity, that is topological similarity between the networks. Here you can see the results. The box plot shows the distribution of the score, the measure calculated between each comparison between package in the 7 samples. And you can see here that we have a high heterogeneity of the results. So we decided to compare the package by using a different approach, by calculating their performance. To do so, we performed for each sample a subsampling, maintaining sampling randomly the 70% of the cells, to then perform again the workflow I've shown you before, so communication, filtering, and then building the communication network, to then compare the results to the one obtained before with the whole data, to calculate true and false positive, true and false, to calculate the performance measure you can see here. Again, the box plot show the distribution of the results for each package in the 7 sample. And here we can assess the good performance of Ulysses compared to the one of the other tools. previously we have seen a high variability of results of the similarity measure, probably due to the different database used as a ligand receptor database. Here we can assess the good performance of Ulysses also because due to the statistical evaluation we provided with Ulysses. Ulysses is not already available. It will be available soon at the link you can see here. We plan to open the GitHub by the end of the year. If you want to be informed about when it will be released, you can contact me or my supervisor at this email you can see here. And finally, I want to thank the organizer for this opportunity and all of you for the attention.
So thank you, Alice, for your nice presentation. And now we're going to switch to James Negai from RWTH University Hospital, whose talk will be about the use of networks for modeling single-cell communication. Please, James.
So let me start. Okay. So first of all, I would like to thank you, the venue and all the organizers. I think it's a big pleasure for us being here, presenting some updates that we are also thinking about Crosstalker. I'm really need to confess that I'm really happy that we are also in line about all the previous speakers have thinking about it, the field. Just a small recapitulation, Crosstalker was proposed in 2021 as a way to compare different settings or different phenotypes using a network-based model. For that, we use the results of ligand receptor analysis, and then we compare those using a different network models. We provide a shareable, reproducible object and report. As most of the talks have touched on it, now we are looking also to intercellular signaling and how does it improves our networks and how important the changes gonna impact our analysis framework. And we do this now by inferring from the single cell data expression. We infer now transcriptional activity using Dorothea, and combining also with a prior knowledge, we can now design and obtain cell-specific intercellular communication links. And it means also that we also have not only ligand receptors in our network, but we also have transcription factors, and therefore we have a much broader network. So here is just small results that we are preparing now. In the left side, you have the old network topology-based embedding, and this is a fibrosis data set that we also make available in your package. And in that publication, we highlight the importance of inflammatory signalings like TGF-beta, collagen 1, fibronectin, because they are really important to the disease setting. But now with your new method, or actually our new network model, we see that those players comes up as more extremely than before. Plus, we also include increase the how much our PCs now are explaining for the disease setting, right? So before you have like something around 70 and now something around 76. This was quite interesting. So yeah, this is just to show how the one of our outliers changed, and also we also can capture what's important and possible transcription factors that are coming up. Interesting, when we address this statistically, it means that we use the ranks of our previous crosstalker, and now we get the ranking of the new crosstalker by gene, and then when we subtract and apply like a rank-based modified z-score test, we see that who is changing the most in terms of rankings is TGF beta 1, which leads us to ask what would be the upstream signaling of it now. And here we also provide a way to trace back which is upstream of TGF beta. So you can see that we have SP1. If you remember, they also appear in your embedding. And upstream of it, we have the receptor integrin ETGB1, right? Here, just the same information that our nodes are projected in the receptor transcription factor edges. And this is, of course, I think, one work that would complement it here is the work made by Luz, where she's trying to really fulfill the activity of receptor and transcription factor. And I think that was really cool, that we are now maybe converging some kind of ideas, right? Interesting, we look into the SP1 row, and we found out this regulates actually maturation of megakaryocytes. And for you that who might not be aware, in fibrosis, in bone marrow fibrosis, one of the cell types that gets really mutated, or they start to be signaling more, is megakaryocytes. And this is also that we are trying to talk with your collaborators to further investigate this in the in vivo lab. Another direction that we are expanding crosstalker, and also was mentioned for one of our previous speakers, is that we asked how we can now understand temporal or time-related cell-cell communication events. So let's say that you have a integrated time single-cell RNA-seq. So here you have a pancreatic ductal carcinoma. Here we have 7 time points, and now we have our crosstalker network. And then our question is like, can we use those network topologies to address or to highlight what would be the early events, or what would be the niche preparation events that could be important to the development of the disease? And to do that, we are applying optimal transport. Optimal transport is an optimization problem that has been applied in single cell in several different problems. And basically, it allows you to transport a given distribution P to another distribution Q. And here, to do that, we get our network. So here you have a single network. We convert this network by using a line graph transformation. It means that here we have cells as or cell groups as a nodes, but when you convert it, your cell groups will become edges. So your nodes will become edges, and then the edges will be connected if you have a shared node with them. And then you can have now from the nodes, or for your edges in the original network, a distribution of signals. And also you can compute a distance using the topology of this new network to obtain a distance matrix. Once you have this type of transformation, you can compute, first of all, compute for all the time points this transformation. And actually, now you can start to compute pairwise distances between cell-cell networks. Another thing that we further want to explore, but we didn't start to look into that is like the possibility to now get a mapping function, and using this mapping function to understand how the signals or how these distributions are transported between small time course events. So to illustrate the distance part, so here you have a pseudo book over this pancreatic murine data, and you see that like here, by using the pseudo book in all the genes, you can kind of retrieve the trend regarded to the time. So here you have time 0. So here's the normal tissue, and here's like a tumoral tissue, and you can see this beautiful time course. And the same time course we can obtain using the Wasserstein distance between the cell-cell network. So you have this from here to here. Of course, further investigations are needed, because like we have this point here that we further need to see why we have this kind of behavior, if the composition of the cell-cell pairs are equal or not, or even what kind of the signals are coming in these two time points to make them closer, right? So of course, as all of the talks have discussed, we have several directions in the cell-cell communication field, and one that makes me really comforted is that we all are recognizing those needs and we are converging. But for crosstalker and also for this approach where we are basically relying on topology and plus expression, of course, because to obtain the topology we get the expression part. We are also thinking a way to use path information instead the intracellular signaling that you have presented here now. I think someone said before that the multi-omics will probably provide a better understanding about the communication, and we also believe that this is going to be happening. So here, once we have a network of ligand receptors and transcription factors now, we would also like to understand if can we maybe use these enhancer regulatory gene networks where people are combining and obtaining from ataxic or multi-omics ataxic RNA-seq to also provide a better understanding about what's the regulation and what's the relation between regulation and communication, and how it can be perturbed or how it can be used to understand complex disease. And of course, for the time series part, to understand these early events, transportation map, which comes in these models, are really a key part. Of course, know how to explore them. This is a tricky question, and this is one of the open questions that we are trying to do right now. Of course, the distance also plays a key role because, like, when you talk about networks, defining distance over network topologies or manifolds are a kind of hot topic. Lastly, I would like to acknowledge again the organization of the workshop, all the talkers. I think I was really happy to see some known faces here, and of course, I was really happy also to contribute a little bit with Daniel. Acknowledge my advisors, Ivan Costa and Michelle Schaub. And yeah, I'm happy to be here and maybe discuss more about it if we have time. Thank you.
Alice, for the last session of contributor talk. So we have 2 minutes and 5 seconds before the session is going to be closed. So I would like to use these few minutes to first thanks the ECCB organizer. So to give us the opportunity to sorry, can I have my screen? Oh, yeah, thank you. So to the CCB organizer, the CCB organization, the BCO Congress that is taking care of all the technical part to make this workshop happen. A workshop that I remember you is organized under the umbrella of New Trends in Bioinformatics by CCB. I would like also to thank the people that today make this workshop working very very smoothly. So Enrica, Luz, and Gabriele that share the session, today's session with me. We have a lot of talk. We have 7 invited talk, 4 contributed talk that I think provide a quite complete view of what it means doing today cell communication analysis from a bioinformatic point of view and using spatial transcriptomic data in our biological and bioinformatics analysis. I think that we have the chance to touch several points, both from the biological point of view, methodological point of view, and have the chance to, consider different aspects. Here are just a few keywords that probably we have the chance to discuss today. But again, area for sure a subset of the topic that today's workshop had the chance to cover. Let me conclude this very nice event with the biggest thanks. So the thanks to our speaker. So let me say thanks to Elisabetta, Lucas, Giovanni, Elena, Eva, Julia, Robin, Daniel, Luz, Alice, and James for a very nice presentation that provide a complete and comprehensive view of state of the art in cell-cell communication and spatial transcriptomics. let me thank even the people that, let's say, behind the scene, design, propose, and make this workshop happen as it looks like today. So Enrica, Lutz, Davide, Chiara, Gabriele, Ian, Julio, and Rosa. Some of them cannot attend to the workshop, but they effort in designing, proposing, and really they want this workshop happen is what makes this event today possible. And the last, thanks, obviously, even considering the light time in the days of dinner time, to all of you for the attention, for attending this workshop. And we hope that you enjoy this event and you find some good topics to do, some good science. So I think from behalf of all the organizing committee and all the speaker, thanks for attending this workshop and have a good night.